Welcome to this explainer. I am so excited to jump into this with you today because we're going to unravel one of the biggest, most fascinating misconceptions in all of modern biology. If you've been following any of the wild breakthroughs in gene editing, you've definitely heard of CRISPR-Cas9. People always call it a pair of molecular scissors, right? Well, today we're looking at the true mechanics behind how this system actually works. And honestly, prepare to have your mind blown a little bit. Because the real editing process, it is way more complex and way more elegant than just making a simple cut.
Okay, let's dive right into this. Because here's the truth that completely catches students and, honestly, even seasoned biology nerds off guard when they first hear it. What if I told you that the famous Cas9 protein doesn't actually edit the genome at all? No, really, it's true. Cas9 has exactly one job: find a highly specific location in the DNA and break it. That's it. It's a demolition tool, not a builder. The actual rewriting, the insertion, the deletion, the true editing part, that is 100% performed by the cell's own pre-existing DNA repair machinery. We are literally just hijacking the cell's natural response to trauma.
So, here is our roadmap for today. First, the cut is just the beginning. Second, NHEJ, fast and messy. Third, HDR, the precision pathway. Fourth, the cell cycle constraint. Fifth, incomplete edits and mosaicism. And finally, number 6, knockout versus knock-in. Let's get started.
Section 1. The cut is just the beginning, hijacking the cell's repair.
The big takeaway here is that researchers aren't really editing DNA directly. They're just deliberately breaking it to trigger a specific biological alarm bell. When Cas9 creates a targeted double-strand break, a DSB, the cell basically panics. A broken chromosome is an absolute catastrophe for a cell. It can lead to death or cancer. So it instantly mobilizes its repair crews. But here's the catch. This means the ultimate outcome of a CRISPR experiment is probabilistic, not deterministic. The cell itself, not the scientist, gets the final say on exactly how that break gets repaired, depending on which repair pathway wins the race to the cut site.
Now, what's really interesting is the grand dichotomy between the cell's two main emergency responders. On one hand, we have non-homologous end joining, or NHEJ. This one is incredibly fast, we're talking minutes, and it works in all phases of the cell cycle, but it is messy and very error-prone. On the other hand, we have Homology-Directed Repair, or HDR. This one is slow, taking hours, and it's highly restricted by the cell cycle. But its accuracy is absolutely incredible because it actually uses a blueprint. Let's take a closer look at the messy responder first.
Section 2. NHEJ. Fast and messy.
If you imagine a broken chromosome like a severed pipe, NHE is basically the cellular equivalent of a panicked DMT wrapping it in duct tape just to stop the bleeding. The molecular mechanism is beautifully chaotic. Literally within seconds of Cas9 making that cut, a ring-shaped protein complex called Cu70/Cu80 spots the broken ends and threads right onto the DNA. That calls in a massive enzyme named DNA PKCS. Then processing factors arrive, including a nucleus called Artemis, which essentially trims or chews back the ragged ends. Finally, a complex with ligase 4 swoops in and blindly glues the whole thing back together. Classic NHE is the cell's primary default, but sometimes it uses other messy variants like microhomology-mediated end joining to blindly stitch things up. And because Artemis trimmed those ends before the glue went on, the repaired DNA is almost never exactly the same as it was before.
This messy repair intentionally introduces small insertions or deletions, which we call indels, right at the cut site. Now, if you drop a random indel into the coding sequence of a gene, you're almost certainly going to cause a frame shift mutation. That shifts the entire reading frame downstream of the mutation, producing a completely garbled sequence of amino acids and, usually, a premature stop codon. So the resulting protein is truncated and entirely non-functional. And here's the thing, scientists absolutely love this messy pathway. It is the perfect, highly efficient way to turn a gene completely off.
Section 3. HDR, the precision pathway. But what if you don't want to break a gene? What if you want to fix a specific mutation or insert a completely brand new sequence? Well, for that, HDR acts way less like a frantic EMT and a lot more like a meticulous architect reading a set of blueprints. It kicks off with a process called end resection, where the mRN complex and CTIP nuclease chew back the DNA ends to create these long, single-stranded overhangs. This is actually the critical point of no return. Once resection happens, the cell completely locks out that messy NHEJ pathway. Next, a protein called RAD51 binds to the single strands and goes on the hunt for a matching homologous DNA template. When it finds one, it invades it, forms what's called a D-loop and uses that pristine template to flawlessly synthesize the exact missing repair DNA.
And this brilliantly illustrates how scientists actually hack the system. Out in nature, a cell uses its own identical sister chromatid as that blueprint. But in the lab, researchers literally flood the cell with artificial donor templates. We can toss in short, single-stranded DNA strings called SONDNs to force the cell to make tiny, super-targeted edits, like fixing a single incorrect letter. Or we can use massive double-stranded plasmids or viral AAV vectors to trick the cell into pasting entire glowing fluorescent tags right into the genome. By handing the cell the blueprint, we get to dictate the exact sequence it writes down.
Section 4, the cell cycle constraint. But as amazing as HDR sounds, it does come with a massive, very frustrating roadblock. You see, HDR is restricted entirely to the S and G2 phases of a cell's life cycle. Why? Because historically, the cell only has that sister chromatid blueprint available after it's replicated its DNA to get ready for cell division. So the cell's internal machinery actively turns off HDR outside of those specific phases. This is a huge problem for modern medicine, because many of the cells we desperately want to cure, like neurons in your brain or muscle cells in your heart, are essentially trapped forever in the resting G0 phase. And in those non-dividing cells, precise HDR editing is basically non-existent.
Because of this natural constraint, there is a literal arms race happening at the lab bench right now to tip the scales. Since HDR gets naturally outcompeted by that super-fast NHEJ pathway, scientists have had to build an entire toolkit to push the cell towards HDR instead. They use chemicals to synchronize the cell cycle. They use small molecules like SCR7 to literally block ligase IV, temporarily disabling the messy NHEJ pathway so HDR actually has time to catch up. They also optimize the physical design of those artificial templates, deliver Cas9 as a fast-acting protein complex rather than raw DNA, and even throw in chemicals to stimulate that Rad51 search party.
Section 5. Incomplete edits and mosaicism.
Now, even with all of those clever tricks, editing an entire organism presents a totally different set of challenges. When scientists attempt to edit embryos, things rarely go perfectly. In fact, they frequently run into a major issue called mosaicism. This is a condition where a single individual develops into a patchwork organism, meaning it's a mix of cells that carry the intended CRISPR edit and cells that somehow remained completely unedited. This was actually a huge, heavily analyzed flaw in the controversial clinical embryo editing case involving He Jiankui. The intended HIV resistance he was going for might have only been present in a fraction of the resulting cells, which completely undermines the entire therapeutic goal.
So, the really crucial point here is understanding exactly why mosaicism happens. At its core, it's a physical race between a rapidly dividing embryo and the sheer speed of the Cas9 enzyme. Think about it. If you inject Cas9 into a single-cell embryo, but that cell naturally divides into two cells before Cas9 has finished finding and cutting every target chromosome, well, you now have two completely independent cells trying to repair their DNA. One cell might use NHEJ and create a deletion. The other cell might not get edited at all. The end result is a fully grown organism with totally different genetics depending on which patch of tissue you happen to sample.
Section 6. Knockout versus knock-in.
Okay, now we can finally tie all of this incredible molecular biology back to the real-world goals researchers have when they sit down at the lab bench. Because the entire field essentially boils down to this perfectly matched dichotomy. If your goal is a knockout, meaning you want to completely disrupt and eliminate a gene's function, you are going to rely on the fast, messy, error-prone NHEJ pathway. It requires 0 templates, works in almost any cell type, and is incredibly efficient. But if your goal is a knock-in, say you want to insert a glowing tag or precisely correct the exact single mutation that causes sickle cell disease, you absolutely must force the cell down the slow, precise HDR pathway by handing it an artificial blueprint. And as we've seen, that is much harder to pull off.
Understanding this really changes how you look at this revolutionary technology, doesn't it? We aren't going in there with microscopic tweezers assembling DNA base by base. We are initiating a highly calculated molecular trauma, handing over a set of blueprints, and just hoping the cell's ancient repair pathways follow our lead. So I want to leave you with this final thought. If Cas9 only breaks the DNA and the cell's own pathways dictate the actual repair, are we truly editing genomes, or are we just brilliantly guiding cells to rewrite themselves? Thank you so much for joining me on this explainer. Keep asking those big questions, and I'll see you next time.